Hello, and I hope this message finds you and your families well and safe. I'm Jim Minow, the Executive Director of the CureJM Foundation. Throughout the COVID-19 pandemic, CureJM has remained focused on our mission to fund research leading to better treatments and a cure for juvenile myositis, and to fund ongoing improvements in world-class clinical care. I want to report briefly on outcomes of two of our projects and their groundbreaking published results, which have just been released. The first is in the field of drug discovery. As you know, it is one of our priorities to identify and make available new drug therapies that could be alternatives to the current standard of care, typically immunosuppressive drugs such as prednisone and methotrexate, which most often have extremely negative side effects. 3 years ago, we began funding a project at the National Center for Advancing Translational Sciences at NIH to identify new drugs with an emphasis on drugs already approved for other conditions. Why those already approved? Because as we find them to be potentially effective in treating JM, approved drugs would be available faster and at far less cost than a new drug yet to go through all of the clinical trials required for approval. This program screened 6,576 compounds, including every approved drug in the US, European, and Japanese drug libraries. The first cut resulted in 216 compounds that indicated efficacy in JM. This first cut was further narrowed to 12 compounds or drugs, most, but not all of which have already been approved by the FDA. These drugs inhibit a pathway stimulated by an overexpression of a protein known as interferon, and the pathway leads to muscle inflammation and weakness caused by autoimmunity in juvenile myositis. You might recognize the names of one or two of these drugs, baricitinib or tofacitinib, for example. Baricitinib has already been tested in a very small number of JM and myositis patients with very encouraging results. The candidate drugs and compounds fall into three broad classes of compounds: JAK inhibitors, non-JAK inhibitors, and HDAC inhibitors. Our goal now is to move some approved drugs into small clinical trials, such as being done with baricitinib, and to move other drugs or compounds into animal trials, which we are starting through a new partnership with the mouse model lab at the State University of New York at Binghamton. The second project I'd like to share with you is a CureJM funded project at the University of Utrecht. This project sought to identify biomarkers associated with a more severe disease course in juvenile myositis. The project was successful in identifying 4 biomarkers where, if expressed in high levels, would indicate more prominent disease activity, a longer course of treatment to reach drug-free remission, and a higher likelihood of disease flare or relapse along the path to recovery. What this means is that the measurement of these 4 biomarkers could aid doctors in making treatment decisions. Patients with high levels of these biomarkers are likely more at risk, and the ongoing monitoring of biomarker levels will help alleviate the threat of imminent flares. Our next step in this study is to validate the results through a larger study. This validation will be important for the widespread use in the clinic of biomarker measurement as a predictor of recurring disease activity and flaring. This US and international research is made possible by your commitment to CureJM. To our longstanding patrons, thank you for standing with us. And to our thousands of families, thank you for your leadership in your communities and for your participation in CureJM's Walk Strong Across America coming up in just a few days on July 4th. There is no question that because of all you do, we will surpass our goal and therefore have the funds we need for the next phases of these and other research projects, leading to better treatments and a cure. Thank you. Please stay safe, and I look forward to seeing you again soon.